Yeah, thanks so much. So, yep, I'm from the University of Wollongong, where I work within chain analysis lab. So what our lab actually specializes in is a technique called mass spectrometry imaging, and especially regarding matrix-assisted laser desorption ionization, or MALDI for short. So what this technique actually involves is we have a glass microscopy slide with our sample on it. This could be a section of tissue or a cell culture. We then deposit a matrix atop this, and what this photoactive matrix allows us to be able to do is we can illuminate it with a UV laser at a very discrete position on the sample. This then creates ablation of our analytes, be it peptides, lipids, nucleotides, etc., and we can then pass this into a mass spectrometer for us to be able to detect. So if we were to repeat this sequence of events after moving the sample plate by a defined position, we're then capable of now visualizing our analytes across a space, so the intensity and then the spatial distribution. So here on the bottom right, I'm just showing a mouse brain, and I've just highlighted two different lipids here with our MSI images. So within the HFSP project, we've really been trying to push the boundaries of spatial resolution capabilities of MALDI. So while commercial instruments are capable of getting down to around about 5 micron pixel sizes, we've now been able to get this down to micron, and in particular instances down to about 250 nanometer. We do this using a technique called transmission geometry MALDI. So instead of focusing the laser through the front onto the sample, we can go through the rear of the microscopy slide, and then this just allows us to be able to avoid the obfuscation of the ion optics and yeah, really focus that laser point down. So now onto some applications here. So we've got neuroblastoma cell line up here in the top left. So I'm just showing that from 5 micron down to 1 micron pixel sizes for 2 different lipids and then some nucleotides here, that we're now kind of capable of identifying organelle type features such as the nucleus in the dark blue there. Same goes with the iPSC-derived microglia. So at 10 micron spatial resolution, we can see heterogeneity within the cell culture, but it's not really until around about 2 micron pixel sizes that we're now starting to visualize the difference of lipids across the cell itself. Just finally finishing on a mouse cerebellum that we imaged at 1 micron pixel sizes. So this is nucleotide imaging here. And the yellow arrows are pointing out the Purkinje neurons there. And we can see a heightened abundance of adenine specific to the cell type here. And yeah, if you want to find out more about what we're doing, I'm at poster 44. Thanks very much.